Hello and welcome to Insight Ophthalmology. I am Dr. Amrit and I welcome you to the part 2 of Aqueous Humor Dynamics. Today we are going to talk about the drainage pathway of the aqueous humor. In part 1, we discuss in detail about the aqueous humor production, and the link to that video will be provided in the description box, and also it will be available on the right side of your screen. In part 2, in this video, we shall be talking about the aqueous flow direction, aqueous humor outflow pathways, physiology of aqueous drainage, the various mechanisms causing resistance in the aqueous drainage, and the response of the aqueous drainage pathway to various pharmacological agents. So let's get started. First, let us discuss about the aqueous humor flow. Since the anterior part, that is the cornea, is cooler due to evaporation of tears from the corneal surface and also because of the avascularity of the cornea, the aqueous convection currents move downwards on the endothelium. The posterior part, we have the iris, and the iris has vascularity, and because of which that area is warmer, and therefore the aqueous humor flows in the upward direction. These convection currents, that is downward direction of flow on the cornea and the upward direction of flow on the iris, is responsible for the deposition of the inflammatory cells, that is the keratic precipitates, on the inferior surface of the endothelium. The aqueous chamber or the aqueous humor's turnover is about 1% per minute. It means that 1% of the total aqueous humor in the eye is replaced every minute. The total volume of the aqueous humor in a healthy adult eye is roughly 250 to 300 microliters, and a 1% turnover per minute means that 2.5 to 3 microliters of this aqueous humor is produced and drained each minute. In 24 hours, this will add up to a complete turnover multiple times, that is about more than 15 times per day. Let's discuss some of the important structures which will be important to understand the outflow system. We shall be talking again in detail about trabecular meshwork, Schlemm's canal, collector channels, and the ciliary body. We already have a video on the channel on anatomy of anterior chamber and angle of the eye. The link to this video will be provided in the description box and also available on the right side of your screen. So let's talk about the detailed anatomy of the trabecular meshwork. The trabecular meshwork is a structure that overpasses the scleral sulcus and converts that scleral sulcus into a circular channel. This channel runs 360 degrees around the iris, and this is known as the Schlemm's canal. The trabecular meshwork is a triangular porous structure that you can see over here in the cross section, and it consists of connective tissue surrounded by the endothelium. Gonioscopically, or from the gonioscopic perspective, the trabecular meshwork is divided into an anterior part and a posterior part. The anterior trabecular meshwork is non-pigmented and is not involved in filtration. It is the posterior trabecular meshwork which is important to us because it is pigmented and involved in pigmentation. From innermost to outermost, trabecular meshwork has 3 parts. These are the uveoscleral trabecular meshwork. In the middle, we have the corneoscleral trabecular meshwork, and the outermost juxtacanalicular trabecular meshwork. You can see that the size of the holes is basically decreasing as we go from inner side to the outer side. The outermost is the juxtacanalicular network or the juxtacanalicular trabecular meshwork. This is in close proximity to the Schlemm's canal, and it has the tightest meshwork out of the 3 layers and also provides the greatest amount of resistance to the aqueous outflow. So we'll talk about that in a while. Let's discuss the inner wall of the Schlemm's canal. This inner wall of Schlemm's canal is very dynamic structure in the conventional pathway of aqueous drainage. The canal is embedded in the scleral sulcus, and it is situated just outside the juxtacanalicular trabecular meshwork. And on the outer side, you can see it is connected with the outer channels or the outlet or the collector channels. The inner wall of Schlemm's canal actually has this ability to form these giant vacuoles. You can see these are nothing but these are infolding of the membrane of the endothelium of the Schlemm's canal, and these will basically pick up the aqueous humor from the juxtacanalicular network or trabecular meshwork, and then they are going to release it in the form of vacuole to the inside of the Schlemm's canal. So this is known as the vacuolation theory. What you can see over here are the giant vacuoles. So these are these large dome-shaped outpouchings of the endothelial cells that protrude into the lumen of the Schlemm's canal. Now, despite the name, these vacuoles are not true intracellular vacuoles. Rather, they are extracellular cavities created when the cell membrane is bulging outwards, leaving a fluid-filled space between the cell and its underlying basement membrane. Now, these vacuoles also change dynamically in response to the intraocular pressure. So whenever there's elevated intraocular pressure, you will see increase in the number and size of the giant vacuoles. And when there's decreased intraocular pressure, a reduction in the vacuole size and number is seen. Another theory states that giant vacuoles per se cannot transfer the aqueous humor, and active giant vacuoles that are transferring the aqueous humor, they actually have pores within them. So pores are micron size opening that can be found within the walls of the giant vacuoles, or they can be found at the borders or between the adjacent endothelial cells. So if they are found within the wall of the giant vacuoles, they are known as the intracellular or the transcellular eye pores. And if they are in between the cells, those are known as the paracellular or the border pores or the B pores. The density and functionality of these pores are again associated with the intraocular pressure. So this theory states that not all giant vacuoles have pores, and only those with pores directly contribute to the aqueous humor drainage pathway. Okay, so this is still a controversial thing. So we discussed about the three layers of the trabecular meshwork, that is the uveoscleral trabecular meshwork, the corneoscleral trabecular meshwork, the juxtacanalicular trabecular meshwork, and then the Schlemm's canal. The Schlemm's canal again has multiple channels. Those are known as the outlet channels or the collector channels. Usually there are about 25 to 30 collector channels that will collect the aqueous humor from the Schlemm's canal. These will drain into the deep and the mid scleral venous plexus and also the intrascleral venous plexus. These plexes are present at the level of the limbus, and from the intrascleral venous plexus, the aqueous humor is going to go into the episcleral venous plexus. Now, sometimes the aqueous can drain directly into the episcleral venous plexus via the aqueous veins. Now, from the episcleral venous plexus, the flow would be into the anterior ciliary veins, the superior ophthalmic veins, and ultimately into the cavernous sinus. As you can see that after exiting the Schlemm's canal, the aqueous humor enters the aqueous veins and subsequently mixes up with the blood in the episcleral veins, where the pressure is approximately 8 to 10 mm of Hg, that is mercury. Now, in case of increased episcleral venous pressure, this will reduce the aqueous outflow, and sometimes, especially in hypotony and in other conditions like Sturge-Weber syndrome, thyroid eye disease, and keratoconus fistula, the episcleral venous pressure can increase so much in comparison to the intraocular pressure that you will find regurgitation of blood into the Schlemm's canal during gonioscopy. So this is known as the blood in the Schlemm's canal. Alright, so now let's finally come to the crux of the topic, and that is the aqueous outflow mechanisms. Since we discussed about the trabecular meshwork, the aqueous humor exits mainly through this trabecular pathway, and this is known as the conventional pathway, responsible for 75% of the drainage. And next we have the uveoscleral pathway, which is the non-conventional pathway responsible for 25%. Basically, what happens in the conventional pathway is that the aqueous humor is going to flow through the trabecular meshwork, whereas in the non-conventional or the unconventional pathway, the aqueous humor bypasses the trabecular meshwork and instead passes through the ciliary body or the ciliary body band. This picture over here depicts the conventional or trabecular outflow pathway. So you can see over here that the aqueous humor is being produced by the ciliary processes of the ciliary body. Then it flows in the posterior chamber, it passes through the pupil and enters the anterior chamber, and from the anterior chamber it enters the trabecular meshwork. From the trabecular meshwork it passes into the Schlemm's canal, and from the Schlemm's canal it is collected by those 20 to 30 collector channels, enters the venous plexus of the sclera, and ultimately into the episcleral veins. From there the anterior ciliary veins, superior ophthalmic vein, and ultimately it drains into the cavernous sinus. Now this pathway is the trabecular pathway and is basically a pressure dependent pathway and is responsible for majority of the outflow, about 75%. Now over here there's a free flow of aqueous up to the juxtacanalicular trabecular meshwork. However, beyond that, the juxtacanalicular trabecular meshwork itself and the inner wall of the Schlemm's canal, they provide some amount of resistance to the flow of the aqueous. This happens because the juxtacanalicular trabecular meshwork is basically has the least size pores and is also referred to as the cribriform layer. The resistance in the conventional pathway tissues is about 3 to 4 millimeters of mercury per microliter per minute. Now here's a clinical nugget. Since it's now known that the major seat of resistance in the outflow pathway is trabecular meshwork and the juxtacanalicular trabecular meshwork and the inner wall of Schlemm's canal, these areas are targeted in surgeries in order to improve the outflow facility in case of primary open angle glaucoma. So we have trabeculotomy and trabeculectomy. Trabeculotomy, we basically open up the trabecular meshwork itself. So as you can see over here, the red line indicates that it has been cut off over there so that you are basically improving the existing pathway of drainage. Now this could be done by various mechanisms, externally or internally, but the whole principle over here is that you are cutting open the trabecular meshwork so that the aqueous flows now freely from the anterior chamber into the Schlemm's canal. In trabeculectomy, what we do is we actually create a separate ostium or opening into the sclera so that the aqueous, instead of flowing through the damaged trabecular meshwork, actually now flows through the newly created pathway. That is trabeculectomy. So in trabeculotomy, we improve the existing pathway, and in trabeculectomy, we are creating a new pathway. Now let's discuss some other mechanisms of outflow resistance as to why does this resistance happens. Now, first of all is the glycosaminoglycans. So in this picture, you can see the structure of the glycosaminoglycan in general. Now, these are an integral part of the trabecular meshwork. These glycosaminoglycans have this property that they can attract water via osmotic forces, and as they attract water, this will cause hydration and edema of the trabecular meshwork. Trabecular meshwork is nothing but holes or those cribriform layers, right? So when there's hydration and edema of the holes or the openings of the trabecular meshwork, there will be definitely increased obstruction or resistance. Now, we do have lysosomes present within the trabecular meshwork, and an important property of lysosomes is that they have these hydrolytic enzyme present within them, which can depolarize these glycosaminoglycans and reduce the obstruction in the trabecular meshwork. And this is a normal mechanism. However, when a patient takes corticosteroids, and this happens more so in patients who are responsive to steroids, so corticosteroids basically stabilize the lysosomal membranes, and these hydrolytic enzymes will not be able to come out. And what happens? The glycosaminoglycan composition goes up within the trabecular meshwork, leading to increased hydration and increased resistance, leading to steroid-induced glaucoma. Okay, so that's a clinical nugget for you. Apart from that, extracellular matrix can also accumulate within the juxtacanalicular meshwork and inner wall of Schlemm's canal, and this is more pronounced in glaucomatous eyes than the age-matched controls. Another protein that has been seen is the cochlean, and this is a multimeric protein, and it's Multiporization is induced by shear stress and other changes in the microenvironment, and this has been seen in glaucomatous eyes again. So remember, glycosaminoglycans, extracellular matrix thickening, and also the cochlear deposition. Is there an influence of iris and ciliary muscle on the resistance to the aqueous outflow? And here we are talking about the conventional pathway that is flowing through the trabecular meshwork. Yes, there is. Now, if you would observe carefully, the anterior tendons of the ciliary muscle, mainly it is a longitudinal ciliary muscle, they get inserted into the corneal scleral trabecular meshwork and the juxtacanalicular tissue, right? So when there's contraction of the longitudinal muscle of the ciliary body, there will be an anterior and inward pull. Anterior means towards the cornea and inward means towards the lens. This anterior and inward pull will open up the trabecular meshwork and dilate the Schlemm's canal. Actually, it's just inner wall of the Schlemm's canal sends in processes, and even desmosomes connect these two structures. So if there's a movement in the trabecular meshwork, the Schlemm's canal is also going to move. So if the trabecular meshwork moves and opens up, the Schlemm's canal also moves, and this decreases the outflow resistance and increases the aqueous humor outflow. Alright, so contraction of the ciliary body improves the conventional outflow or the conventional pathway. Whereas if there's relaxation of the ciliary muscles, in particularly the longitudinal ciliary muscle, there will be narrowing of the trabecular meshwork, and this closes the Schlemm's canal, and there will be increased outflow resistance. So this completes the conventional outflow, and now let's talk about the unconventional pathway that is responsible for about roughly 10 to 25% of the aqueous humor drainage. Here, as you can see, the aqueous humor is being produced again by the ciliary processes of the ciliary body. It passes into the posterior chamber. Posterior chamber is the part of the anterior chamber behind the iris. All right, so it passes behind the iris, through the pupil, and then you see the dashed line starting, right? So what's happening here, that instead of flowing into the anterior chamber, the aqueous humor now gets absorbed within the interstitium of the iris roots and the ciliary body, and it passes between the bundles of the ciliary muscle and goes into the supraciliary or the suprachoroidal space, and from there it gets absorbed by the veins which are present in the choroid and in the sclera, or sometimes it even passes through the scleral pores directly into the episcleral tissue. This is not responsible for the majority of the aqueous humor outflow, and therefore this is known as the unconventional pathway. This is also a relatively pressure independent pathway, and it contributes to about 10 to 25% of the outflow. What is pressure dependent or pressure independent? It basically means how much a system or an outflow is dependent upon the intraocular pressure. The uveoscleral outflow decreases with age, and this kind of balances the decreased production of the aqueous humor with age. Also, meiotic agents can decrease the uveoscleral outflow. We'll talk about this in a while. Uveoscleral outflow increases with prostaglandins, with cycloplegia, cyclodialysis, and increased episcleral pressure. Ultimately, the entire aqueous is draining into the episcleral pressure, so into the episcleral veins. So if there's an increase in the episcleral pressure, obviously what's going to happen? The outflow is going to go down, whether it is conventional pathway or it is the unconventional pathway. Cyclodialysis is what? It is separation or dialysis of the ciliary body from the sclera, right? So when that happens, obviously the aqueous which is passing within the interstitium of the ciliary body, from where does it get absorbed? It gets difficult for it to get absorbed into the sclera and into the episcleral tissue. So cyclodialysis also leads to a decrease in the uveoscleral outflow. So again, this is a summary of the uveoscleral outflow. You can see the aqueous humor exiting via the ciliary muscle interstitium, supraciliary space, and then into the sclera vortex veins, and finally into the cavernous sinus. Ultimately, everything drains into the cavernous sinus. Alright, so now let's discuss about the uveoscleral pathway. How is it dependent upon, how does it changes with the contraction and relaxation of the ciliary muscle? Now, by now you should be very clear that ciliary muscle contraction increases the trabecular outflow. But what about ciliary body? the aqueous humor is passing within the interstitium or through the interstitium of the ciliary muscle, right? So if there's contraction of the ciliary muscle, that interstitial spaces will the space will become narrow, and therefore there will be decreased uveoscleral outflow. Ciliary muscle relaxation, on the contrary, and therefore cycloplegia, is going to increase uveoscleral outflow. It has been seen that in eyes treated with atropine, uveoscleral flow accounts for about 4 to 27% of the total outflow. But when you give pilocarpine, which is a miotic agent or a cholinergic agent, the uveoscleral outflow will come down. So here's a clinical nugget for you. Usually we know that miotics increase the conventional pathway or the trabecular pathway outflow. Alright. However, in case of traumatic glaucoma, or in case of angle recession, or when there's significant damage to the trabecular meshwork, it's a uveoscleral outflow which becomes the major pathway because trabecular meshwork is already damaged. Now, in these cases, if we administer miotics to the patient, there will be contraction of the ciliary muscle and there will be decreased uveoscleral pathway. That means we will be compromising the only pathway which is working for the patient, that is the uveoscleral outflow, and therefore there will be a paradoxical increase in the intraocular pressure in these patients. Now let's discuss about the prostaglandin F2 alpha analogs like the latanoprost. Now they increase the uveoscleral flow because they act by remodeling the extracellular matrix of the ciliary muscle to reduce the resistance. Then we have another important equation that is a fundamental formula used to describe the dynamics of aqueous humor in the eye and to estimate the intraocular pressure. And this is given by intraocular pressure equals to aqueous humor production rate minus the uveoscleral outflow divided by outflow facility plus episcleral venous pressure. The outflow facility, you can equate it to the trabecular meshwork outflow. So it can be written like this, where F stands for rate of the aqueous humor formation, FU stands for uveoscleral outflow, C is the outflow facility, basically the ease with which the aqueous humor exits through the trabecular meshwork in microliters per minute per millimeters of mercury, and PE is the episcleral venous pressure. However, this is not without limitations. It assumes basically that the uveoscleral flow is pressure independent. Now, what did I tell you? That it is relatively pressure independent. Okay, so it's not strictly true that it is entirely pressure independent. Moreover, it simplifies that the episcleral venous pressure, as a single value, even though it fluctuates. So it's a range 8 to 10. But this equation considers it to be a single value, right? So those are the limitations of the Goldman equation. Let's see how the aqueous humor basically is regulated along with the cardiac cycle. Now, in diastolic phase, that is when the heart is relaxing, the aqueous is pumped from the anterior chamber and reaches up to the Schlemm's canal. But the main drainage, that is from the Schlemm's canal to the aqueous veins, occurs in the systolic phase when the pressure within the heart rises or when the heart contracts. Alright, so we cannot end this video without discussing the normal range of intraocular pressure. So the normal range of intraocular pressure is 10 to 21 millimeters of mercury, right? And if someone asks you about the average intraocular pressure, don't get confused. The average means that mean plus and minus standard deviation. For a general population, that would be 15.5 plus or minus 2.6. And the normal range is basically plus or minus 2 standard deviations. So if you calculate accordingly, you will get normal ranges 10 to 21 mm of mercury. With that, we wrap up the Aqueous Humor Dynamics Part 1 and Part 2. If you haven't seen the Part 1, the links are right there in the description box. Make sure to check them out for a complete understanding. If you like this video, you probably enjoy our videos on anterior chamber anatomy and gonoscopic anatomy as well. And of course, don't forget to like, share and subscribe if you haven't already. It really helps us continue bringing high quality ophthalmology content to you. For those who want to support us further and access exclusive perks, the membership link is also in the description. At the end of the description, feel free to check it out.